Good evening, students. Today we will talk about lipids, right? I'm going to revise lipids today. So, you can also take out your NCRTs, mark the important lines, and you can understand lipids with me. First time, the term lipid, it was given by Blood. It was the scientist, basically Bloor, who introduced us with the term lipids. Mostly students, they feel very, kind of, they are not able to define lipids exactly, that what are lipids. They confuse with fatty acids, they confuse with oils, they confuse with what is ghee. Then there are lots of terms regarding to the fats and oils and lipids. So many times we are not able to differentiate that what is the difference between them. So now listen to me very carefully, that the simple general definition of lipids is that they are esters of fatty acids and alcohol. Means they are formed by esterification. It means two important components if you want to form lipids. What are those two important components? Fatty acids and alcohol. Join fatty acids plus alcohol. Which bond do you need? Now if I say join, it means you need some bond. You need ester bond. So which bond will be formed when you join fatty acids with alcohol? Your ester bond will be formed. So here you can see we have taken here our simple lipid, that is with the glycerol. We have taken here CH2OH, CHOH, CH2OH. This is our glycerol. And functional group with the carboxyl group COOH, that is our fatty acid. So here we are making triglyceride, so we will use 3 chain of fatty acid COOH. Then there will be third functional group COOH. Now when these 3 fatty acids, they join with this glycerol, how many water molecule will be released? 3 water molecule. Which kind of synthesis it is? Dehydration synthesis. Through dehydration synthesis, we are going to form our simple lipid, that is triglyceride, right? So basically which bond is getting formed? As you see there, we are removing this. So which bond is getting formed here? As you can see now, so our ester bond is getting formed. So you can make your simple lipid. So lipid is what? They are esters of your fatty acid plus glycerol, or there can be any alcohol. They are hydrophilic or they are hydrophobic. They are hydrophobic. They are hydrophobic. We will talk about, definitely we will talk about amphipathic molecule also, which are your phospholipids, which are both hydrophobic and hydrophilic, but lipids are usually hydrophobic. They are water insoluble. Now they are soluble in another non-polar solvents like benzene, like ether, like chloroform. So now we are going much little deeper into the lipids. We are clear with the definition of lipids? Very much clear. They are formed by two things. Fatty acids को join करो alcohol के साथ, you will get your lipids. Now one type is that very simple lipids, which we just talked about, fatty acid with alcohol. One is little complex, compound lipids. And third is more complicated, that is derived lipids. So we have 3 types of lipids: simple, compound and derived lipids. Don't worry, we will discuss one by one that what are these and what are their examples. Simple lipids, we just defined that they are fatty acids plus glycerol. Their esterification means when they form ester bond, then they are known as simple lipids, right? So triglyceride, if I ask you now, you can confidently say, ma'am, triglyceride. This is example of simple lipids, right? This is a simple lipid. Fats and oil. Now to means they fats and oils, they are your natural lipids or true fats, as well as they are our simple lipids. Now, so many times we say that these fats and oil, they are basically our triglycerides. They are triglycerides of fatty acids and glycerol, which are made up of with one glycerol head and 3 fatty acid chains. But why? Now how you will differentiate between oil and ghee? What is the difference? Oil, when we say oil, it means those fats which are liquid at room temperature, they are known as oils. So they will have high melting point or they should have low melting point. Means they will have low melting point. So oils, they are liquid at room temperature and their melting point will be low. Then question comes is, what are fatty acids? Fatty acid is those compounds which are having one functional group joined with carboxyl group, right? COOH. So basically, when you hydrolysis of fats, then they form, this is our fat. So when hydrolysis of fat, they will form your fatty acid. Now I am hydrolyzing this triglyceride with the hydrolysis. Hydrolysis ke liye what do I need? I need water. How many molecules of water? 1, 2, 3. So 3, and I will get my fatty acid. So fatty acid is fatty acid is component of lipid. Lipid is made up of 2 components. What are those components? Tell me quickly. Fatty acid plus, what should I write? Alcohol, right? And simplest fatty acid, here so we are using 3 fatty acid chain. Otherwise the simplest fatty acid we know is formic acid, HCOOH, simple carboxyl group attached with your hydrogen. That is the formic acid. Now fatty acid, they are further of 2 types. I did not say now lipids are 2 types. Lipids are 3 types. Which one? Simple, compound and derived lipids. Fatty acids further of 2 types. You must have done in chemistry that there are chain which are having sometimes single bond and few chains which are having double bonds also. So those fatty acid which do not have any double bond between carbon atoms, they are known as saturated fatty acid. Example is like palmitic acid. It is 16 carbon. It is having 16 carbon, as well as butyric acid. Unsaturated किनको बोलते हैं हम? Those which are having 1 or more double bonds in their chain. If anyone is having 1 double bond, it is known as oleic acid. Linoleic is having 2 double bonds. Linolenic acid, it is having 3 double bonds. Arachidonic acid, it is having 20 carbon, including the carboxyl carbon, it is having 20 carbon. And there is another 2 important things, MUFA and PUFA. MUFA is mono unsaturated fatty acid, those fatty acid in which only 1 double bond is present. right? And they are usually healthy fat molecules. They basically they keep your tummy full for a longer diet, and they are good to eat even basically they should be in your diet, in your daily diet, because they help to lower the cholesterol also, both mufa and pufa. Nuts, nut butter, oils, olives, avocados, dark chocolate, they are good healthy fat molecules, we call them. And they are generally liquid at room temperature. It means their melting point is lower. Same is for pufa. Pufa is a very healthy type of fat which is liquid at room temperature, and it is very well known to help the cholesterol. It is having omega 3 and omega 6 fatty acid which you get from fish, from nuts, from corn oil, from sunflower oil, as well as there is a one flaxseed oil. Flaxseed oil, that is also basically very rich source of your omega 3. One another thing, linolenic and arachidonic, they are very very essential fatty acid which should be included in the diet compulsory, right? So it doesn't mean कि fat सिर्फ बुरे होते हैं. We need at least 20 to 30% fat, healthy fat in our daily diet. So basically polyunsaturated fatty acid, monounsaturated fatty acid, they are very good fatty acid, but saturated fats, they are not healthy type of fat. Basically especially some fats in the few meat, as well as few oil, few dairy products, they are having saturated fats, and they are not as healthy as pufa and mufa. They are trans fats which are your present in burger, fries, as well as pizza. They are not good for you. That is why they are also, they all raise cholesterol level in the body, and they are in the category of fast foods. Now comes third one is waxes. They are also another simple lipids having a high melting point. High melting point, they means they will be solid at room temperature, insoluble in water. In plants even there is a waxy layer which help to reduce the rate of transpiration. It makes the basically plant tissue waterproof. And there is a wax, basically simple lipid which is present in blood also, that is the cholesterol. As well as another very very well example is bee wax. Actually bee wax is a simple lipid. It means made up of two things, fatty acids and glycerol. So it is made up of palmitic acid and alcohol. So it is another common example of simple lipids. Now comes to compound lipids. Fatty acid तो होगा ही होगा, glycerol will be there, but additional group or element is there. On the basis of the group, they can be of different types like phospholipids, glycolipids. Phospholipids means they contain phosphoric acid, and phospholipid is an amphipathic molecule which is both hydrophilic and hydrophobic cellular parts. They are present, the phospholipid which is present in our cell membrane, very 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 important point. Phospholipid which is present in our cell membrane is lecithin. And soybean oil also is having phospholipid, that is cephalin. Another which is present in our nervous tissue, it contains carbohydrates, it contains fatty acid, it contains glycerol, it contains nitrogen, that is glycolipids. It is present in the white matter of nervous system. The third and the last category is derived lipids. Never you will find them in a straight chain. We call them lipids without straight chains. You break the simple lipid, you break compound lipids, you will get your basically derived lipids. So like sterols, they are having very complicated structure, long rings, long side chains, like your cholesterol. From cholesterol, it is a very good source for bile salts, for making steroid hormones, for the formation of vitamin D. We call it is a main precursor for the formation of salts, steroid hormones, and vitamin D. Another, which is derived lipids, which are present in our oils, essential oils, which give very strong aroma, very beautiful aroma. Camphor oil, eucalyptus oil, lemon, mint, जिसमें से खुशबू आती है बहुत ज़्यादा, terpenes. And another which is present in your pigments, phytol is also derived lipid. It is a, basically we call it as a terpenoid alcohol. It is, which is very rich in vitamin A, vitamin K, vitamin E, and also present in pigments like chlorophyll and carotenoids. So basically 3 types of lipids we talked about: simple and compound and derived, right? Best example we talked about is the cholesterol here. And then we talked about that the fats and oils, basically if the melting point is low, if the melting point is low, they will be in the form of oil. But usually saturated fatty acid, they are having high melting point, so they will be solid at room temperature. Go through this this lipid structure as well as lecithin structure from your NCERT, and I will revise DNA and RNA, nucleoside and nucleotide.